Welcome to February Fridays. On last Friday in April, we're talking Pennycress today. We've got Julia and Matt talking about a reading update on Pennycress, and then we have Fariba talking about an update on soybean cyst nematode associated with Pennycress. We'd like to acknowledge our sponsors that make some of this work possible. The Environment and Natural Resources Trust Fund, the Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, Builders Vision Philanthropies, and the Ignite Foundation, among other generous donors. We should start, as we always do, with any announcements. Mitch, do we have announcements from February?
Yeah, we do. So I'll start with happenings. On Tuesday, we had a meeting of the Winter Camelina 1 million acre study, 1 million acre scaling study, which is OneMass is the acronym for 1 million acre scaling study. Partners Circle. So that's kind of a mouthful, but this is our effort to develop a shared cross-sector roadmap to move from where we are now, about 5,000 acres of winter camelina in the region, to 1 million acres. So it's pretty ambitious, but it's, the idea is that if you're going to do that effectively, you need to be coordinated across sectors, know who's doing what when, what investments are needed when, et cetera. And so we've got a team of consultants helping to lead that analysis. And then we're having these quarterly partner sort of meetings. So we had, 40 or 50 people in a conference room in Wasika at an AURI facility. And we heard some updates on analytics or the analysis. We saw a camelina seed cleaning demonstration by one of our farmer partners, Paul Novotny, who's built the seed cleaner. We also saw camelina being crushed by AURI at their facility, and that was really cool to be hands-on with camelina meal and see the oil collecting in a bucket and all of that. And then we went to Rosemont and stood in a 65-acre camelina production field that is set up for relay planting with skip rows. Looks really good. That station had pretty good luck with relay planting last year, and so they're trying it again. And at that location, we had 5 or 6 different Minnesota congressional offices, staff members represented, which was really cool to see policymakers recognizing the potential around winter camelina and coming out to learn. So Colin is leading that effort for our team. So huge kudos to Colin, but a big team effort to be there and contribute. That was great. And then also this week, in a different part of the Forever Green universe, we finished planting our first hazelnut Go First farm. That's at Kimber Contours down in the Farmington area. Les was a big part of that, and the whole hazelnut team, plus some volunteers.
Including his father.
Including, I wrote my dad into doing it, and he was very excited about it. And I think is low-key considering whether he wants to put in hazelnuts at his farm someday. So we'll see about that, but yeah, that got completed. That's been, I think, 3 years in the making, less. So just based on plant availability, it took us 3 years to pull together not just enough plants, but exactly the right plants and the right genotypes to fill out each row. Each row is a distinct genotype, so that's going to be really cool to see as they mature, see the differences and their unique character come out. And that's a massive accomplishment. So another big thing this week. 2 more things I'll add. One is that we made it through the chalk talk. So the co-directors are all, we had about 45 people participate in each one of those chalk talks. So there's a lot of time devoted. I think 25 or 30 people came to all 3. So a huge thanks to all of you who took that time and also gave us your feedback. The feedback has been incredibly helpful. Search committee has met. We're moving the process forward. The potentially disappointing news is that I think it'll be a little bit before we can announce who it's going to be, just because we've got to work through some more final details here. So don't hold your breath too much, but we are making good progress. And the last thing I'll say is that we also on Tuesday at that million acre scaling study partner circle meeting, we had the first onboarding day of our new winter oil seeds specialist, Maciek Kazula, who's online here. I'll ask him to say hi in a second. Maciek was thrown right into the maelstrom there, not only attending a massive meeting with all the partners who are going to be critical for his job, but even being asked to facilitate a breakout and present on that breakout on his very first day on the job. He did great, of course, and we're really excited to have Maciek. So Maciek, do you mind giving us a couple sentences or a few minutes, a minute or so with your background, I guess?
Thanks, Mitch. Yeah, hello everyone. Happy afternoon. I'm excited to be part of the Forever Green team and to represent the winter oilseed portfolio. I look forward to bringing my expertise into this role, growing through this transition and become eventually your go-to resource over time. So I'm especially eager to collaborate with all of you and learn from your expertise starting from today. I'm sure you're gonna have plenty of opportunities to getting to know each other. I don't see you now over the camera, but I hope there is a full room of people in the room. And yeah, look forward to catching up with you soon in person.
Awesome. Thanks, Maciek. And I'll just, for context, of course, we have our big, winter oilseeds research team, but Maciek's role is going to be to help weave all of the pieces together because what we're doing ultimately touches science, touches the economy, touches policy, and keeping all of that coordinated and also driving forward rapid progress is getting to the point where Colin and I can't, keep it all together anymore on all those levels. And so we really need some more dedicated support, and that's what we'll have with Maciek. So thank you, Les. There's another upcoming opportunity to volunteer to plant hazelnuts Saturday at the Jokulus farm. I think it's called Sogan Valley.
SOGN, Sogan.
And that's in the Cannon Falls area, right, Les? So not a far drive, and I think the weather's supposed to be pretty nice. So that's tomorrow. Saturday's tomorrow. So if you're free tomorrow morning or during the day tomorrow, it'd be great to go help out with that. And if you're interested in participating, you can email Matt Levitt, who's often on these meetings, Leavitt. He's your contact person to get signed up to volunteer.
And Mitch, is one mask just for camelina, or does it touch on other?
So good question, Jim. We debated a little bit in the beginning whether it was camelina and pennycress or just camelina. I was more on the side of let's cover both, but I think the broader partnership felt like that would get a little too complicated, kind of keep both crops in mind. So more similarities than differences, but especially on the regulatory front, there are some significant differences.
Oh, yeah, you did all.
But the way I think about it is that the camelina plan will cover 80+% of the challenges and barriers we've got to figure out for Pennycress. So it will pave the way and enable Pennycress to follow in those footsteps. But definitely, it's only really focused on those two oilseeds. That being said, this general approach of taking a very concerted, analytical, and cross-sector partnership-based approach kind of ahead of time to think through a scaling plan and then implement with partners is something that we, Forever Green leaders, want to replicate. And it'll probably look different for different crops. I mean, we're spending quite a bit of money, and luckily have good foundation support to run this for camelina. Of course, there's a ton of attention on camelina right now, so that's how we generate that. It may look different for a different crop, where we're kind of DIYing it a little bit more, but regardless, you think that this discipline and this approach has a lot of potential.
I'm going to give an update of the Pennycress breeding program, particularly focus on the domestic performance of domestic lines. And after a talk, and Matthew Otter, who is the assistant breeder of the oilseed breeding group projects, will talk about GWAS mapping of soybean cyst nematode resistance in Pennycress germplasm. I'll briefly start with the team who did all the work I'm talking about, and then also like to give a history review of the taxonomy of the Pennycress, where to start, where it was, where it is now, and then until breeding program overview, and talk about the domestication gene introduction and then the performance of the most domesticated lines. Here's the team. And this, the people you can tell who is doing the work. Right now we have 5, including me will be 6 people. That's Serena, Sarah, and mainly focus on the Pennycress. But we do a lot of work together. And Matthew is in this combine here is we're focused on Camelina, and then the field technician and Chris is 50-50 between the two crops. And Chris is a grad student slash technician. And I have to talk about, here is another, the situation we are embedded, we are in Dr. Jim Anderson's Spring Winter project. We get a lot of resources sharing and help from his project. I said how many people are in here, how many people it takes to build a moisture curve for Pennycress. If you count, there's the number 5 here in the complex Susan, that's the weight recognition here. And then one person I like to talk about is Dr. David Marks. He developed the domestication genes for our to work with. Also, we have a partnership with Cargill. And this is a picture of our last fourth meeting in Cargill headquarters. Pennycress, the scientific name is Telaspi avance. I try to be sound as Latin as possible. They are both winter type and spring type, but this annual, the yield potential is about 1,000 to 2,000 kilogram per hectare. And then the most research study said that at 1,500 kilogram pounds per acre is a profitable crop. The oil content is about 30 to 36%, and mostly the market will be biodiesel and sustainable aviation fuel. You heard a lot of SAF. It stands for sustainable aviation fuel. The protein content is pretty high, and it's a similar level as canola. I show a picture where the Pennycress name comes from because the pot size is similar size as penny. The history of the SLASP taxonomy SLASPD is the tribe. It has been very confusing. I want to emphasize what we have found. Initially, in the literature, you said there are 76 SLASP LS, which is the last sensor lateral broad-sense species. In later 76, start with the SLASP, but in 1976, a scientist, he split this into 12 genera. And then one thing I want to talk about, the heavy metal accumulation species was placed in 2 different genera. And in fact, this is a tool. In fact, the most recent study said that they belong to different tribes. They don't belong to the SLASPD, but the problem is we misuse a lot. People don't catch up with most recent study. Don't agree. This is a chart showing. This one is a use of publication year and the number of publications. The blue is keep using the old updated name. We continue. If you look at it here, continue. Because Pennycress getting attention, SLASP studies began to have a lot more. But by the 2000s, literature started to use the correct taxonomic name, began to pick up. But majority is still wrong name. And also in the 1990s, the color is molecular marker gene sequence data also support this. So the only 6 in the 1970s study, only 6 original 76 SLASP species was placed into the SLASPD tribe in 1970 and 1970. And they keep going in when the DNA technology came, they're more interested in to clear up the confusions. So in 2018, and then the SLASPD was split into 14 genera. I'm not talking about details. In the SLASP, there are 4 species left only, where the avance, the field Pennycress belongs to. But in the same year, another group of scientists did more research and they went back to the original collected specimen in the Herbaceum and connect with where those species come from, get a new new samples, and then the conclusion is the Sloth-P, there are only 3 species left. This is where Sloth-P avans. That's our field Pennycress. There are 2 others. But and then the confusing why you have 4 left here, because the most recent herbaceum specimen said those 2 actually are non-English names. It's just a quality of the original specimen were poor, so it's classified different species. Here's a nice picture. We talk about Pennycress don't have diversity, but that's a really interesting picture of the diversity of the Sloth-PD tribe. You see the color variation, there's a purple color, the fruit type look different. This one of them look even like, to me, like cactus on the rock. So that's all that's always a fun thing to do to follow your wild relatives, even you are crop scientist of your domestic crop. Okay, any questions here?
No, that's super cool though.
Yeah, I think I talk about this is because in the science or anywhere the name matters. And then we need to clarify what we exactly have. Pennycress Sloth-P once. It has a typical character for rapid genetic improvement, and including starting by domestication. It's self-pollinated, it's very simple diploid, only have 14 chromosomes. Genome is very small, and it's genes are single copy, easier to manipulate. And the life cycle is relatively short, particularly with spring type, where it's 3 months you can from planting to harvesting. This is a picture I found in the field, like this one, the butterfly on. The breeding target and the first thing numbers, one thing we have to domesticate it. The critical domesticating traits we need, I'm working on uniform seedling emergence and reduce the pod shattering and also the early maturity. This is very important if we use this as a cover crop or intermediate winter oil crop. So have an early maturity pennycress, we can plant do an intercrop, integrate pennycress into intercropping. And also we have been experimenting to plant a summer crop after pennycress is harvested. So far, result is positive. As far as the agronomy traits is the yield, including the seed yield and then the oil yield per acre. And it's always a balance of yield and maturity. You want early, very frequently got to compress the yield. And the winter hardiness and standability, this is by plant height and origin, and the seed size is useful for planting. If your seed size is bigger, you have better soil seed content, and also easier to cross in the crossing facility. The end use quality is oil and meal quality. Is the oil trait. I'm not going to elaborate. And then one thing we are working on is Dr. Mark's create a mutants create edible oil, which is low uric and high oleic. And the meal quality method is low glucosinate content, high protein, and low fiber. This is the breeding. How I run my breeding program. On the left side is the elite breeding program. We make crosses, particularly with the wild types. And also after I do a first general yield trial with something promising, and this elite lines will go to a backcross breeding program. After backcross twice, and then we are going to, this is called domestication lines, and go back to the yield trial and consider the elite potential variety candidates. And then both of them, this is two different breeding programs if you have a hands-on experience dealing with backcross. The domestication genes we are working on, this is Dr. David Mark's creation. Those are the 5 genes and I'm breeding with, and those are mature, I think, and he did have a lot of data to back in. back the utility is of those trees. The number one is called is a certain resist gene. This is a TT gene, transparency Tesla. That's a seed coat color is this. That's a yellow seed, and also the fiber is low. Another two, the Phi and the rod is a low erucic and high olive oil content. And then the third one is the last one is a low glucosinate gene. That's right now we have is UP2, but we have a two backup genes and then to deliver these trees. So the breeding target is largely driven and sometimes is influx is depending on the market demand and what market segment go. Like the the sustainable aviation fuel only need a wild type oil, so that we don't need the two oil chains, which is the fatty acid, the low erucic and the rod gene. But for the whole grain, which mostly now the carbohydrate is using to feed the boiled chickens, is need a low erucic. You don't need this rod gene. But for the edible oil, we'll need both of those. Mutants of both genes contain the 50 to 60%.
Just one clarification, because
Yeah.
it took me a little bit when I came into this job to understand this for non-geneticists. The capital letters of the genes are the wild type,
Yes,
and the lowercase letter is the mutation, is the mutant version. So
yes.
that's why it's the same letters are listed for all three, but different capital and non-capital letters.
Yeah, thank you. We initiated the back crossing of the 4 domestic genes to 5 elite lines in 2022. We are started and then we finished. And then this is the timeline. I'm not going into details. You look at it, you have F1 to backcross F1, backcross to backcross 2. And then they have to self the back BC1 F1 to get F2. And then total cost, lot the data marker point we did, I calculated about 35,000. And then the last fall, 2024, the fall of 2024, that's the first year I planted those the F3 seed or back cross 2 in the yield trial. So it took 3 years. That's why I think I'm very careful when you start this, which one you want to work with, to be selective, to know what you are looking for. And then, so this is scatter plot of the cast markers we use to select for those trees. And then because one thing is the frequently in Pennycress randomly I see the unpredictable and non-mundane inheritance. So to make sure I can recover those gene combinations, I work out the target is 4x of a normal mundane inheritance. If I do start with 4x and I have good success, get the heterozygote of all those 4 genes. So the work is more than what we did, what scientifically is in your practice. So for example, BC1, the 4 gene is 16. You only need a 16, and I did 16 by 4 is 64, right? We have to make a lot of crosses, get 64, at least 64 F1C. That's the idea. And then this is what you would see in the cast marker if it's back cross F1. If you did everything right, segregation, you only have 2 clusters. This is wild type, this is a heterozygote type. And then back cross F2, we'll have 3 clusters. We select this one. In F1, back cross F1, we select a fixed, the homozygote mutant in F2.
You said it's unpredictable and non-Mendelian, but what you just described to a non-geneticist makes it seem like it is behaving the way that we would expect.
So the ratio is not
the
right.
Ratio, that's wrong.
Yeah, but you never would be able to 3 cluster here if it's F1, back cross F1.
Okay, so it's the ratios that show up.
Yeah, that's a ratio. That's why our workload takes much more effort to make even the crosses. for Pennycress. You have to make sure your marginal area is much large. If I fail in backcross F2, I have to start with start backcross, start my F1 again.
Yeah.
Yeah,
Right.
okay, thanks. And then this is, if you look at this is just our quality control, the Y type. Here is the where we grow those materials, and then we have growth chamber, put in little tree, and then those leaf punched, and while those FB backcross plants are being harvested, each one is tagged. We are genotyping the lab, they are being vandalized, and after that, they are transplanted in the greenhouse and big crosses. There's a lot of cross bags, and that's how they look like in the greenhouse when we try to blow up the seed in the F2 stage. The yield trial of the last year is planted, I selected 200, about 250 lines of those 5 recurrent parents or elite lines. And genetically, they can have 87% of genome of the donor parent. And also want to use the opportunity to study the genotype by trait, which is domestic genes and the interaction of those things too. And I'm not going to go to detail. So I have 4 experiments. Those 250. The first one is the 4G all fixed homozygote. Those have a seed for 4 reps. So I split them in 2 locations, Wasika and St. Paul. The second one, AYTC2, is fixed on the essential genes, the shattering and the yellow, but they could be fixed one of the other genes or heterozygote. Also have seed for 4 reps and planted in the St. Paul and Wasika. The PYT, this is PYT1 is enough seed for 1 rep because those greenhouse harvested. And the carotene I have a location there, just it's I planted some undomestic material there because it's black there to look at for seed increase and also compare the yellow and the black effect. So, but that year was very, remember 2024 is extremely dry. Any location we didn't have irrigation was discarded. And then carotene, we had good luck. And those 2 locations here, this is, we are not going back anymore. That's historically where I have been planting Pennycress. And the yield trial, the trees I collected is 4 emergence. And normally this 4 stand is collected 2 or 3 weeks after planting. And in the spring, assess the winter hardiness and our stand score, because the Pennycress wild type, you see germination in the spring even. And then the performance wise, last year, the first flower happened in April 29th, and then the earliest of inheriting was 2 weeks later in the May 7th, which is the first part of May. And in St. Paul, the earliest matured in June 16th, the middle of June. And then carotene was before the 4th of July. And then we do collect some phenotype data. I still look at the shattering score very critically. And then post-harvest, we will run the seed in the Marvin and in II. And the opportunity traits, last year, the best opportunity trait is erosion resistance. This whole field, this is a field of how Pennycress looked like last year after 2 very harsh storms. And then if you look back, this plot is all down, but some of the breeding lines, domesticated lines stand very well. This is comparison, how the domesticated lines perform compared to each of the domesticating parent, the recurrent parent or the donor breeding lines with the recurrent. So this is last year data. If you want to have an average, this is the recurring parents average is 350 kilogram per hectare of oil, but the average of the backcross domesticated 620. And then the seed yield, the undomesticated version is 1,000, but our domestic line, backwards line, was about 700 kilograms higher than per hectare than their recurring parent. And then also we increase oil content very substantially by 4% overall. And the standard score, the fourth standard score is 7 is very good already. So, and then I couldn't see this clearly. So the highest increase is oil yield is about is 176% of the domestic, the breeding line. The yield, seed yield increased 60%. Another one, stand score is increased substantially, and oil content also increased. You saw that, and then the positive. Oh, another, of course, shattering score here is reduced. It's no, seed size was reduced. It's smaller than the wild type. And then very funny thing was the plant height stayed the same, branch height went up, and the panicle length shortened. I was just to see if morphologically we are changing too. Okay, that's the first year data. One question we have been debating what to do with those two oil genes. This is a low erucic gene, FIN1, and the rod and the FIN together would give us edible oil, high group. But here is, you need to think about it. This is domestic pennycress percentage of the donor, the wild type. So wild type to wild type is 100%, right? This is the basic breeding parent, the donor parent. So the blue color is the Phi gene, erucic gene. That's we have been talking a lot. Same as there's no penalty on the yield, on the Phi. And then if you look at follow, but the oil content in the Phi is reduced by 5%, reduced percent of the wild type, the mutant type, and also the oil is oil content reduced here, this one. So because the yield increased so much, and then the total oil yield was not statistically different. And then the stand, somehow there's a penalty on the false stand while we use the low heuristic acid mutant. What else? And then there's a little bit later, of course, a little bit later maturity, and maturity rate, flowering and maturity rate, and plant height shortened, and the panicle length shortened. So overall, FIN1 reduced oil by 3%. If you use percent not to compare, and then it's have a negative effect on the stand. The pink orange is the rod gene, and the only negative thing I thought is cause the maturity a little bit late than the others wise as a relatively mutual mutant. So you can have it, you don't have to have it.
One of your strategy objective is to increase the seed size.
Yeah.
So when I see on one of your slides, you increase the oil content, does the seed size decrease? Are they mostly dependent?
I think here is the data, oil content and the seed size, right? So this is not always. So for example, this one we have oil content 39. If you look at this one, the seed size of this one is 1.6. So if you compare with mean one was 6, because everyone is lowered, but if you compare with mean one was 6, and it's still bigger than mean one was 6. I think we increase seed size in our elite donor, and by that we gradually increase the seed size of the next generation of domesticated lines, even you compare with the wild type, is still low.
The first step is that when you incorporate TT, it makes the seed smaller.
Yeah, that's what TT
So that's
does.
the big difference.
Yeah,
That's why all those lines on the bottom are in green for seed size.
yeah.
But then through breeding, we can boost it back up.
Yeah, I think if you cross those lines together again and again in breeding, I hope by part of the increase of yield is seed size, I believe, is it come together. I have not, yeah, so that
Yeah.
makes sense. But I think we did make progress. This is the meanwhile 6 is the domestic version seed size is meanwhile 6 when you convert to domestic line. The seed size also dropped substantially. But we are still better than meanwhile 6. Okay, this is what else? Questions? Talking about. I'm talking. Amazing performance of twin domesticated Pennycress lines. This is the name. We began to have a Pennycress, P7 Pennycress mean number, and it's easier now rather than a long line. And then all the seed color yellow, and then the wild type is black. And then this is a market segment. This self is and wild, and then this is whole grain, and then this edible oil. So the highest yield we can get is 200 kilograms per hectare, and then if you compare with our best meanwhile 6, we still increase the yield what 700 kilogram per hectare. It's pretty. I'm happy about it. Even if you can't get the oil, because we increase oil content percentage even higher by the oil yield. Also, there are some lines mature earlier than the meanwhile 6, like this one, MU 25014. It matured in St. Paul is what 6 days sooner data to maturity than the meanwhile 6. I hope once we get out this fitting island, maybe it's. And also there's a variation in the glucosinate. Glucosinate is a trait we are working very hard and giving us hard trouble. But in the new collection, new domestic lines, there's some is low, it's like this. In the same field, and then we have some, the highest is 130, and then there's some is 60, is about 60%, 50% less than the highest percent. By selection, maybe we have a chance, just by regular breeding selection, we are lower the glucose content. The rest, I have some lines that start to increase seed. Top 5 of the top performing lines are being increased in the field in D1, right along Carpenter. The one by the road is a seed increase plot. You will see some of the early flowerings already flowered a lot this month. It's a busy slide. I just show you some genetic variation and the progress we have been made. What's going on? What's going on in the field which we planted last fall? And then most of the yield trials now are derived from the backcross program. So I have 4 locations, yield trials, 3 locations are non-weed. So we can go back to the field a couple of years later, this is for Vasica keratin. All the station managers are very happy to hear that. I'm more welcome. And if some experiments are in 3 locations, some lines I gave to yourself for phase growing that in 7 locations. I also have a seed increase. That's how it looks like in the fall. That's a D1 field. This is Carpenter, right? That's Cleveland on this side. One thing we are proud is the existing harvest bags are too small. I took a picture last year. I didn't know what's coming, but I thought it's good news. I just took a photo of this is our parent, and that's where we are last year. So it's a full bag. And then in the fall, we talk about it. Sarah also brought that issue that she thinks we should increase seed size, the bag, it's time. So we are ordering the bigger harvesting bag. Visual of this data. Not everyone is like that, but the top performers are like this.
Thanks, Julia. Yeah. Fariba online is going to talk about the phenotyping end of it because that's a lot of hard work that she did. So thanks again, Fariba. And I'll just talk about the GWAS side, trying to find markers associated with the trait of soybean cyst nematode suppression by pennycress, by different pennycress accessions in lines. There were, in our analysis, 387 that had both phenotype and genotype data. So using those 387, just included the genotype and phenotype data, and I tried various GWAS kind of softwares, programs, and I'll get into that in a little bit, but it turned out that one of them performed better than the other, and I'll say why in a little bit too, but firstly, just came up with this table. I'll explain this table a little bit. When you have, in statistics, if you have multiple comparisons, then there's a good chance that you're going to get like a positive result just by chance. So a false positive. And so we have hundreds, thousands of comparisons with genotype data. It's kind of a balance. You don't want too many markers because then your statistical power goes down because you correct for the multiple comparisons. So if you can have fewer multiple comparisons, you can have a little more power. But then if you don't have enough markers, then you're going to maybe miss the marker that makes the most difference. So that's the reason I kind of did this in an iterative approach. So I downsampled the total marker data by 30. So 30% of the total, 40% of the total, 50% of the total. And so when you're downsampling, you use different randomizations to do that downsample. So those are called random seeds. And so just in case, like maybe by chance you missed one of the key markers when you downsample, I did a few different types of random selection to maybe capture something that was missed. So I did 3 different random seeds and the 3 different downsample amounts. And then there's another thing that's about principal components, like how many to include in the analysis. And that helps the software avoid the confounding results from population structure. So these populations of pennycress are collected from around the world. They've been bred a little bit, and there's non-random association between markers and traits, not sometimes just because they're not fully mixed. There's this population structure. So you have to account for that too. So so I have, I'll get into that a little bit more too, but basically I did 27 different analyses, 3 times 3 times 3, to kind of find out what maybe were the strongest QTL, and just kind of came up with a score based on like how many QTL show up in that relative position based on these different iterations. So that's what this table shows. The strongest reliability score, I call it. It's maybe not the best name for it because it also refers to something else, but we can think through names later. But is chromosome 6 and chromosome 7. Pretty strong on chromosome 4 as well in the middle. So that's just kind of a little background on the table. So that Bonferroni threshold, that's the significance threshold. That's based on the number of comparisons. So you take the number of comparisons, and that's in the denominator of alpha. So if your alpha is 0.05, you divide it by the number of comparisons, and that's like your Bonferroni correction. So that's why you kind of need a balance between like too many markers and too few markers. So here, this is the full set of markers, over 26,000 markers. And that not only was it too many, I tried it with fewer markers too, but we still weren't getting like anything that was significantly over that threshold. So that was with the program called Sommer in R. So I tried it in Tassel, but I think, yeah, if you could go to the next slide.
Okay.
Similar thing, result in Tassel. But someone in Jim's lab, Harsimar, recommended I try a program called RMVP, and that uses software called Farm CPU. And Farm CPU just basically reduces the number of false negatives, but also the number of false positives, just by using an iterative approach to eliminate the confounding between population structure and kinship. So if you go on the next slide, that Farm CPU worked for us with RMVP. So this is just like one of the 27 iterations here. So I just like kind of made a tally, like, okay, chromosome 4 left arm, chromosome 5 left arm, chromosome 6, 7, and just made a tally of all 27 iterations. And then that table that was in the beginning, that's where this came from. So yeah, we're looking into the gene space right now. We've got some Cas markers designed to kind of look for and validate these QTL. So actually Senyu and Fariba were asking me today about some questions about validation, so we can validate them too. So thank you again, Senyu and Fariba, for helping with the validation of these. That's also more work. So yeah, thanks for that. Is there another slide?
Yeah.
Oh yeah, and this is just the scree plot that is a way of determining like how many principal components to use in the RMVP software. So the first principal component explains most of the variance, 60%. And then you just, I did 3 to 5. You just kind of, this, like, I put a link in here, like what I followed, the recommendation to do that. So I just kind of drew basically a vertical line, and I used 3 to 5 principal components in that analysis. So that's where that comes from. I think that's it, right? Is there any, yeah, that's it. Yeah.
That's my last slide. I started with the team, I end with people. Again, I think thank you very much for everyone in the team. And then I call our team resilience. Today I gave my name. I think we work with something we new, and then we very frequently and unexpected hit a wall, but we always be able to find the ladder and climb over the ladder. This example, what I want to show here is what I thought after first planting. We had some seed. We got a new planter. Everybody worked hard, found out how to use that planter. But I thought, what's going on? There's some plants in between the alley. And then the whole team worked together, found out what's the problem. And then continue this spring, and we got the winter tiger people come, and then that's what we found there. Inside the cedar, in the winter tiger planter, there is a, what do you call this?
Yeah, it's inside of a pen. It's like the ink part of a pen.
Yeah, ink part of the pen. Oswaldo noticed that.
Were you tearing it open, or how did you actually find that?
We were all there.
Yeah. He was, yeah.
Yes.
Was opening it up. No, actually, I put it in.
We were kind of testing part of it.
Yeah.
Yeah.
Yes.
Yeah, I was testing it out, and I realized it.
You noticed it, yeah.
Wow.
So we hope this. We have other things in place, but this is a mechanical thing. It's what you do when you. And then lastly, when I look at this bag of seed, I'm always wondering what Don would see if he were here.
Have you established minimum trait values for each of the traits before you would allow it to go to commercialization?
I think that's a good question. I think number one, that's what we said, is what's the profitable for yield, right? Number
For
one,
each trait?
for each treat.
Yeah. What's the minimum? Do you have a minimum value for each trait before you would allow that line to go commercial? Oil standability, all your traits. If you've got minimum values established before you would allow it to go commercial.
I think, yeah, that's a really one difficulty question is what's your baseline, right? Even you are doing a new crop, but for me, it would be the minimum of 6 domesticated. It's very good. Even the wild type, sometimes it's just always very good. That would be our baseline. Once we get a new germplasm coming in, maybe we raise the bar more.
Yeah, and
Yeah.
practically speaking too,
Yeah,
like, it starts out small, you
Yeah.
get the farmers who are really passionate. And they'll test things on small acres that, they know there's some risk there. But if it works well for them, it's like, it's almost more of like the farmers kind of tell us, like, is this good enough for us? And they're like, if they plant more, then it's good enough for them. So that's part of it too.
yeah, it's a two-way traffic. We have to listen to them too. Yeah, yeah.
But you don't want to put something out that's gonna bomb, because then you get a bad reputation.
Yeah.
Thank
So
you
the
for
question
the
is, what's the minimum
advice.
for each of those traits before you allow it to go commercial?
Yeah, no, for sure. I think,
I think a good advice.
and that's why it's like starting small too is good, because then you can test it. It's like, is this gonna fail? Is this acceptable? Does this farmer like that? Is it meeting Cargill thresholds? Maybe, that kind of stuff. Yeah, Cargill maybe would set some of those minimums too.
I think it's interesting how different it is compared to an established commodity where with an established commodity, it's a high bar to show that your new variety is better than what's out there. But at least you know exactly what you're comparing to. Here we're comparing to, something that's being made out of whole cloth. So I think you could do an economic analysis, right? Of oil yield per acre could be kind of the overarching limitation. If you don't reach a certain potential oil yield per acre, that could be profitable, assuming that we're pushing us in the direction of a fuel crop. That could be the key minimum, while at the same time not having weediness and having very minimal shattering and very minimal lodging issues, right? It has to work for a farmer in those ways and also give a yield that can work economically. But we're also, as we're thinking about how does this roll out, or just looking at what Covercress is doing, they're starting with a chicken feed product that isn't trying to meet a particular oil yield per acre. They're getting their feet wet and learning how it works on farms in a very cautious and de-risked way for farmers so that you don't have that bad reputation. So like that principle, I think, is something that we feel really strongly about, especially future releases. I think maybe we didn't understand that well enough when we put kerns out into the world, just to be frank, right? We have a lot of folks, including a lot of farmers, have taken some lumps in rolling kerns out. And, I think we're going to learn from that going forward. But, I think it's going to depend on what that end use market is, how you define the minimum threshold. But I really like the point, Les, that we should be really focused on that. I agree. That's a very smart way to think about it.
Yeah, it is. Yes, it's a good thing is we make the rule. Bad thing is we don't know what's a lot of them. It's just we make it while we are practically working on it. Just something is acceptable. Yes, I like here in my breeding program. I look at this maturity. We started here. Anything if mature at the same time, or even mature a week earlier, but the yield or oil content the same as meanwhile sick. I call it if you made the progress, right? You made the maturity earlier few days. You maintain the same yield of where my baseline.
Yes, I want to know if it's possible to compare this with canola. Yes, to establish the threshold of yield and in oil content to see if this is profitable.
So that's what the most recent research is here. This is somebody did the economics, because you also have to look at what's the input, what's the oil price. When you sell as a sustainable aviation fuel, what's the price, current market price, and what's the meal price? Those are all to be determined. Even this number is here, maybe someday, but this is I think somebody based on the canola model. Yeah. This is published data at the site.
Hello everyone, my name is Fariba. I'm working as a postdoc at Senyu's lab about the nematodes. Today I'm going to talk about two projects related to the soybean cyst nematode and interaction with the Pennycress. So the first project I want to focus on overwinter survival soybean cyst nematode in pennycress, roots, and soil. And the second one project is screening more than 400 pennycress germline and breeding lines to SCN that Matthew talked about the GWAS study about it, and today I'm going to talk about the phenotypes data. So first of all, I would say about a little bit about the soybean cyst nematode. The soybean cyst nematode is one of the most important pathogens affecting soybean production, and as you can see the picture of right side, this pennycress, if this soybean cyst nematode life cycle, it started from the egg, and then after the hatching, second stage of juveniles, as an important actual and main stage of the nematode can effective on the roots, and then second and third stage hatching in the roots, and then the nematode is maturity to the female and the male. Male actually sometimes remove the roots, but the important stage is a female and make like lemon shapes with a lot of eggs on it. So it's maybe 200 to 300 eggs containing to the cyst, and again it started this life cycle. And the pennycress is then Julia talked a lot of perfectly about this crop. So it's a winter annual crop that can growing during fall and early spring. Because of that, this is maybe act as a alternative host to the SCN in the during of winter. So however, we still do not fully understand which stage of the soybean cyst nematode can survive winter condition, especially in North America. So studying overwinter survival in pennycress is important for developing better management strategies. So in this study, we established different soybean cyst nematode development stages inside pennycress roots before winter. We inoculated the pennycress with the second stage of the juveniles and allowed them to development from different days, keep in the growth room for 2 days, 5 days, 9 days, and 14 days before transfer them to the field. So this allows us to have a different stage of the juvenile, different stage of the nematode from juvenile second stage to development stage like third stage and fourth stage of juveniles and adult. So we also inoculated some soil of treatment with second stage of the juveniles without any plant such as pennycress or soybean, and then moved them outside to the field. So in this picture, we first planted the pennycress in the growth room, and after 10 days to 2 weeks, we keep in the growth room and then inoculated with the second stage of the juveniles, and then keep for 2 days, 9 days, 5 days, 9 days, and 14 days in the growth room, and then move them outside in the holes in the field. And for each treatment, we have 3 plots. And then we have a different time point sampling. We bring samples inside and check for survival or yes or no. So for this study, we have repeated this experiment for 2 seasons and 2 years to ensure a consistency of the results. So after overwintering, we evaluated SCN survival in 3 ways, actually. So the first, when we want to understand the nematode can be survived or no, or which stage can be survived, we bring some pennycress inside at different time point sampling. For example, prior to freezing, after soil thaw, 1st of April, and different times. So bring pennycress inside, wash the roots, staining the roots to observe developmental stage of the nematode in the roots. And also, we transplant pennycress into the new cones to evaluate whether nematode remaining viable or can be reproductive the nematode or no. And also the third one for understanding the nematode can be survived in the soil or no, we measurement cyst formation in the soil after bring cones inside and plant soybean and keep for 35 days in the growth room and check the cyst formation. For the first year, the results show that soybean cyst nematode second stage of juveniles could survive in soil during winter field condition. So prior to freezing and after soil thaw, when we bring some samples and plant soybean in the cones and keeping them 35 days in the growth room and wash the roots and collected the cyst, we can found the cyst formation in the cones. So that means the soybean cyst nematode, second juvenile could survive in the soil and also can be actually development different stage of nematodes, and we can have a cyst formation. And what about the roots? The pennycress roots, the nematode can be survived or no? So for this, we have two ways, transplant, and also we check the roots with staining. So prior to freezing, when we bring some pennycress inside and wash the root and staining, we can detect it actually different stage of the juveniles in the roots from second, third, and fourth states. And after soil thaw, when again bring some samples inside and wash the roots and staining, we can again detect it different state of the juveniles in the roots. And but 1st of April, when bring some samples inside, we can just detect it second stage of juveniles. It means that this time the nematode cannot be survived. But for the transplant, unfortunately, for the check the viability, when transplant the pennycress in different time point sampling prior to freezing, after soil thaw, and the 1st of April, unfortunately in the growth room, the plants, some of them already died, or also some aphid infected in the plant, and we have not limited data about this one. So the roots show that second stage of juveniles can be survived and development to the third and the fourth stage, but we have no idea the SCN can be reproductive and remain in the roots or not. And this picture I can show you about the different stage of the juveniles in the root from the actually this time is a prior to freezing, and we can detect the different state of the juvenile in the root. After soil thaw, we again wash the roots and the staining, we can detect the second stage of the juveniles, third or fourth stage of juveniles in the roots. And also the 1st of April, we just detected second stage of the juveniles in the roots. And what about this year? Until result now show us that the second stage of the juveniles could survive again in the soil during winter field condition as the same last year. So prior to freezing and after soil thaw, when bringing some cones without any plant inside and plant the soybean and keep in the growth room for 35 days, we can found the cyst formation in the soil. So it means the second stage of juveniles can be survived in the soil without any plant during the prior to freezing and also after the soil thaw. But what about the plants? What about the pennycress roots? Again, we have two methods to actually measurement the nematode can be survived in the pennycress root or not. Again, we staining the roots and look at the roots in the for the actually detect the different stage of the juveniles, and we can stronger nematode development was observed period of freezing, especially for the third and the fourth stage I can found in treatment 3 and treatment 4. So treatment 3, it means we keep the pennycress in the growth room for 9 days, and treatment 4 for the 2 weeks in the growth room, and then move them outside. So in addition, when we transplant some pennycress in the new cones and keep them for 35 days in growth room, I can detect it, actually we can detect it, cyst formation in the roots of the pennycress prior to freezing. But after soil thaw, when bring some samples, again we can detect it second, third, and fourth stage of the juveniles in the roots. But for the viability and actually transplant, unfortunately, again, treatment 3 and treatment 4 our plant already died. So we think about it maybe for the temperature outside this year, and is compared to the last year is temperature, some days very high and some days very low temperature, so up and down. So we have effective to die the plants, but when the treatment 1 and 2 were healthy, and then bring samples inside and transplant them in the new cones, but after 35 days we cannot detect any cyst formation. But then the 1st of April again bring samples inside and check the roots, and we can detect it second, third, and fourth stage of the juveniles in the roots, especially just in the treatment 4, but the other treatments we cannot detect it. And for the viability, again now independent in the growth room, and maybe first week of April, I want to be check them and look at the treatments. But as I say, it's already treatment 3 and treatment 4 died plant in the field. So just I transplant treatment 1 and treatment 2 for the 1st of April and then keeping them in the growth room to check them. So here again, we detected different stage of juveniles from the 2nd stage to 3rd or 4th stage of the juveniles before the freezing, and also after soil thaw. Again, we detected different stage of juveniles, and also for 1st of April in treatment 4, we can detect it the 2nd stage of the juveniles to 3rd and 4th stage. So also here pictures I can show you when we bring some samples, Pennycress inside and transplant them. These are pictures from the period of freezing and 25th November 2025, and keep them for 35 days in the growth room, and I check them, and we can detect it, cyst formation in the treatments period of freezing. But after soil thaw, also you can see here, when I want to transplant the Pennycress in the new cones, the roots are, some of them plants already died in the field, and some of them, when I bring inside and wash the roots, you can see the plants roots are brown color and damping off. When I transplant them in the growth room, after 2 or 3 days, they are damping off, or you can see here the picture, they are not very healthy. So about it, we have a missed data about this part. So overall, I can say that across the 2 years until now, the results show that SCN can survive successfully over winter in both soil and the Pennycress system, especially in the most consistent in the soil as a second journal says. However, Pennycress roots can support nematode development stage from the 3rd, 4th stage in the roots, but as I said, whether SCN can reproductive or not, the transplant them is still, we have limited data about this part. And so finding our results suggested that Pennycress can act as a trap crop and maybe as an active role in maintaining SCN population in soybean production systems. And the second project that Matthew talked about, the GWAS studies, we have more than 400 germline and breeding lines of the Pennycress on to the SCN HG type 0. So we planted the Pennycress and again inoculated the plants with the eggs of the nematode and keep in the growth room. And after that, we check which lines can be survived, which lines can as a susceptible, or which line as a resistant or moderately resistant. So for this objective, we need to be collected the Cs, count the Cs, and actually calculate the female index. The female index as calculated the number of the female of each line is divided by number of the female on the soybean susceptible soybean lines, and then times the 100. So the numbers show us that if female index is below the 10, that we can say these lines are resistant, but it's female index between 10 to 30, we can say these lines are moderately resistant. But when the female index is below the 10, is higher than the 30, we can say as these lines are susceptible. So our results here, when we look at the female index, there are very variation in the Pennycress lines, but just we can detect 20 lines are below the 10 female index. It means these lines are resistant, just the 10 lines, the 20 lines, sorry. But the other lines, most of them are as between 10 to 30 as a moderately resistant, and some of them are higher than 30 of female index. It means susceptible lines. So it means show us a very variation of the actually the lines. So this result can be show us we can have a detected genes and QTL of the lines have a resistant to the SCN, to the new future breeding programs of the Pennycress. So when I end off my talking, I want to say thank you to my supervisor, my collaborators, Senyu and James and Julia and Matt for help me a lot about these projects, and also technicians help us to the research to the Pennycress study with the interaction with the nematodes and the different projects. Thank you so much.
The first study, I was trying to figure out why the plants were looking dead in the spring. And then in one of the last slides you had on that study, it showed the fall, how they looked in the fall when you were evaluating them prior to transplant, I think. On the top left corner, are those plants that were later transplanted into the field?
Top left, these pictures, maybe?
Yeah.
Yeah, this picture is prior to freezing when bringing some samples inside on 25 November. And yeah, keeping the growth room for 35 days, and the plants were healthy and without any damaging for the roots or everything, and we can detect its cyst formation. But after soil thaw in the March and also in April, also we didn't have any good healthy plants in outside the field.
Looks to me like those plants have bolted in the top left and therefore probably lost their winter hardiness. So I don't know if these were spring types, because I think you may have looked at
The
springs
type.
and winters, or
We planted 106 variety of the soil. The Pennycress is a winter.
It's a winter
It's a winter, yeah.
it's all min 106.
Yeah, all of them 106 is a winter.
I'm wondering if those plants were vernalized in a cold chamber before they went outside, because they really shouldn't bolt like that in the fall, right? Other Pennycress people?
Not meanwhile 6.
No.
Even you plant in late August, you won't see bolting until spring.
So, I mean, I don't think it invalidates the fall data at all, but it just might help us understand why we didn't see viable plants in the spring.
Yeah, they were growing outside. Is a cool condition. We thought we may not need to vernalization it before. We maybe treat it with a cool temperature before we move out. But it seems we really need.
I think I think maybe we're misunderstanding each other. I think it would be a bad thing if you had vernalized them before putting them out, because that would cause them to bolt like this. So you're saying that you did not vernalize, is that correct?
Right, yeah.
So yeah, it's a little bit perplexing as to why they would bolt in the fall if they're a winter type that wasn't vernalized.
Did they make seed, or were they making pods at all, or flowers?
Yes, it's make seeds for these parts, yeah.
Oh, they are. So yeah, I've seen some things bolt, at least in Camelina, without fertilization, but they don't make flowers.
I never seen that happen in Pennycress.
So yeah.
We planted Pennycress. Dr. Diefenbachs planted his Pennycress last year before Labor Day, around the Labor Day, right? They didn't see that happen. Unless it's springtime. Springtime does bolt in.
Yeah.
If you plant too early, you will see that happening.
I have a quick question on the slide with a bunch of bar graphs.
This
like,
one?
well, I mean, I should say a bunch of bars on one graph. The 425. So how are these ordered? Because it seems like there's a little bit of pattern. Yes, from like 97 to 241, it looks like a steady increase in female index. So did you kind of order them somehow on the x-axis, or?
From these parts, it's a high female index.
Because that's the line ID. So line ID 97 through line ID 241. It's just like almost like a linear increase.
Mm-hmm.
So is that by chance, or is that ordered that way somehow?
I don't think so. It's when this is a high number of the female index. Yeah, it's chance. Do this randomized? I don't know. It's
Fariba, I think this number I thought is order of the numbers. Is not the line numbers. I believe you did it this way. And probably one group is winter, and another is spring lines, and then there's some is mixed.
Yeah,
Maybe it's not really line ID. It might be just from low to high.
No, this is not a line ID. This is just the order of all the numbers.
Gotcha. Okay. Yeah. Thanks for answering. Yep.
Yeah. So these are mixed from different spring, winter, and mixed pennycross.
So this is your own internal number, right?
It could be.
Yeah.
You experiment with them, however you label them. So I got a question for Fariba. What those collections from the wild? These are we call the nature collections. What's the variation between the plants within each line? For example, you tested the plant number, Pennycress number 1. Within this line, what's the variation? I assume you tested more than one plant. What's the variation between those plants?
There's 7 reps, right? 7 reps.
6
A lot of reps. 6, was it 6?
6,
Okay,
right?
yeah. 6 reps.
Yeah.
Maybe you just didn't add the error bar, but there were a lot of reps.
Yeah.
So, yeah.
These are 6 reps, 6 plants. Yeah.
Okay.
Yeah, I think that would be really interesting to know how consistent and repeatable are these results. I think in the past, when some of this work has been done, we've seen a certain line look pretty resistant in one study and then look pretty susceptible in the next study. And so having all those reps should help us sort that out if there is anything repeatable and therefore genetic about.
Looking at it, it looks like, there's a lot of lines, a lot of data, but generally speaking, just looking at the ones that were, most resistant, it looked like it was pretty good consistency too. So that was a good sign for me to keep, pressing forward with the genotyping work and stuff, the mapping. Yeah.
Yeah, I think like the resistant ones with very low score, if you have some plants is really high, that'll be a good opportunity for us even keep the low ones as a seed source in breeding.
Yeah, as a good parent. Yeah.
Yeah.
Yep. So we can identify those parents, not just the markers, but the parents too.
Right.
Yeah.
This might not have been fully apparent, but the idea is that we have this phenotype data and the GWAS that Matthew ran based on this phenotype data.
Yep.
Right? Yep. So that the two together will help us not just identify lines, but potentially identify genes that are related to that.
Yep.
Existence.
Yeah.
So,
exactly.
Yeah.